Welcome to another episode of Progress, Potential, and Possibilities, discussions with fascinating people designing a better tomorrow for all of us. I'm your host, Ira Pastor.
Welcome, everybody, again to another episode of our show, bringing you another really fascinating guest today, helping to create a better tomorrow for so many people around the world. We have the opportunity today and the honor of being joined by Dr. Jerome Kim, who is Director General of the International Vaccine Institute, which is a nonprofit international organization. It was established back in 1997 as an initiative of the United Nations Development Program, dedicated to the discovery, the development, and the delivery of safe, effective, and affordable vaccines for global public health. They're headquartered in Seoul, Korea, South Korea, hosted by the Republic of Korea, 36 member countries, and the World Health Organization on its treaty. Dr. Kim served as the principal deputy and chief of the Laboratory of Molecular Virology and Pathogenesis at the Military HIV Research Program at Walter Reed Army Institute of Research. In addition to being the project manager for the HIV vaccines and Advanced Concepts Evaluation Project Management Offices at the United States Army Medical Material Development Activity. Dr. Kim was a professor within the Division of Infectious Diseases of the Department of Medicine at the Uniformed Services University of the Health Sciences. And from 2004-2009, he led the Army's Phase 3 HIV trial for RV-144, the first demonstration that an HIV vaccine could protect against infection, as well as subsequent studies that identified the various laboratory correlates of protection and sequence changes in breakthrough HIV infections after vaccination. Dr. Kim graduated at Yale University School of Medicine, completed his training in internal medicine and fellowship in infectious diseases at Duke University Medical Center, received the John Maher Award for Research Excellence at the Department of Medicine at Uniformed Services University of the Health Sciences, and throughout his career has published over 200 peer-reviewed publications. We're honored to have him today. Dr. Jerome Kim, thanks so much for taking the time to come on our show.
It's my pleasure. Thank you.
It's starting out. I spent some time, Dr. Kim, going through your extensive publication record, and you have published extensively across the infectious diseases. You've published on HIV, on tuberculosis, in some lesser-known areas. The first oncogenic retrovirus, T lymphotropic virus type 1. You've published in some obscure microbes, Pseudomonas putrescens. Infectious disease clearly became your sweet spot. Talk a little bit about how that all started and how that happened.
It's kind of interesting because you never actually end up doing what you originally set out to do. I think when I was at the University of Hawaii, I wanted to study regeneration, which has always been an interest from AP biology class in high school. And there wasn't anybody at the University of Hawaii who was studying regeneration, but the advisor for the biology program said, "Oh, you should talk to Mike Hadfield. He's looking at marine flatworms, which I guess are capable of regeneration."
Oh, yeah.
And so they showed me a tank, and Mike looked in and he said, "We don't know what that thing is." And that was the start of my career in research, doing an analysis of an unidentified marine flatworm in the tanks of the Kewalo Marine Biology Laboratory of the University of Hawaii. I mean, not what I was intending to do, but it was, a good introduction to, the kind of patient work that goes into observing, an organism that hadn't been described before, all the things that you needed to do in order to classify it, which was the first question. And I learned a lot about microscopy and how to do thin slices. And then one of the graduate students helped me with an electron micrograph. So basic introduction to the techniques, and you know, it wasn't exactly where I wanted to be, but I did learn a little bit about what was needed, the kind of patients and activities that went into the work that you do when you're trying to study a particular problem. And then when I went to Yale, I said, oh, hey, this is really great stuff that they're doing now, sequencing and understanding how antibody genes are put together. And, you know, it'd be really great if I could. I would really love to do that. So I went to a counselor again, and they said, oh, well, you know, well, your antibodies come from B cells, and we have a B cell immunologist at the Howard Hughes Institute at Yale, Kim Bottomley. So why don't you talk to her? And so I spoke to Kim, and I didn't end up doing, you know, sequencing or understanding how antibody genes were working, but we decided, she put me on a project that you know, actually helped to describe the different functional classes of what we call T helper cells, which we now know actually affect the way antibodies are made and the types of antibodies that are made, and you know, was one of the earliest demonstrations of that. And you know, this is a medical school thesis, so I worked the summer of the first year, the summer of the second year, as much as I can during the third year, which is our clinical year, and then in the fourth year, I took off as much time as possible to work in the lab. And it was really, you know, working with Kim Bottomley, who eventually became, you know, the president of Wellesley, was really an introduction to science and how you do science. And the immunobiology group at Yale included some very well-known figures, and it was a really amazing opportunity to learn about how science is done, not necessarily, again, studying what I wanted to study, but you know, really something that really became a passion. And then, you know, when I went to Duke, HIV was, you know, just kind of getting started. And, you know, we didn't see as many patients in North Carolina as I saw in New Haven. But it was still something that was really fascinating. And of course, you know, you go to talk to the head of infectious diseases, and the first thing he says is, well, you know, this is a dying field. We should really think about doing something else. So I started to fill out those things of, you know, applying for cancer fellowships. And as I was looking through, I thought, you know, I don't think I could do this. I mean, as interesting as cancer is, and with all the advances that have been made in the last 40 years in cancer research, it could have been a right decision, but I looked at it and thought, no, you know what? I really want to do infectious diseases. And so he said, okay, fine. We'll take you into the program. And there's this new guy who just got to do Warner Green. He comes from the NIH. He works on T lymphotropic viruses and HIV. And that's how I ended up doing that work. And it was a good introduction to HIV, because when the Gulf War started and I wrote to the Air Force and said, you know, can I defer entering because I just got another grant and it would be great if I could finish up this work. I got an email back, who are you and why don't I know where you are? We need to bring you in. And so they sent me to work at Walter Reed Army Institute of Research. I was in the Air Force. I was the only Air Force person there. And it was in a group that was doing HIV vaccine research under a guy named Bob Redfield, who eventually became the head of CDC, and then under Debbie Birx, who then became the ambassador for PEPFAR, the global health ambassador in general, and then went on to, you know, advise Trump about COVID. And so it was really a great group of people, and the work that was done was really important in understanding how to prevent HIV infection with immune responses driven by vaccination. So it's a long way of introduction to, you know, you don't always work on what you think you're going to work on, and you don't always get to do what you want to do. But in the end, a lot of questions in science are very, very interesting, and, you know, taking one and learning a lot about it, and it does become something that it's hard to put down.
Absolutely. That's fascinating. So here you are now. And as I mentioned at the beginning, IVI is involved in all areas of the vaccine spectrum, from design to development to field evaluations. And you know, just looking at the sort of the partner section on your website, you have such an impressive list from the global health organizations, the World Health Organization, UNICEF, CEPI. You have the pharma groups, Merck's and Pfizer's of the world, the Gates Foundation, Wellcome. The list really goes on and on. Also with this goal, as you define on the website, of the diseases of the most impoverished, of the DOMI. Can you talk us a little bit about how all this got started in terms of, because the scale and the scope of what you're doing there in South Korea is so very impressive compared to a lot that I've seen. Talk a little bit about how this all got going, specifically on these diseases of the most impoverished.
And so, yeah, this is you know, it's a very inspirational story. But a group of people got together in the late 1980s and early 1990s, and with funding from the Rockefeller Foundation, they founded the Children's Vaccine Initiative. You know, so at that point, advances in vaccines had really brought a huge kind of a revolution, and you know, there was a great consensus around the benefit of vaccination in children, something that we really need to reestablish. And it was really, you know, this group of people. Who included some, you know, real luminaries in vaccines and vaccinology, people like, you know, Hilleman and Henderson and Phil Russell, who used to be in the Army also, by the way, and the late Phil Russell. And they said, you know, we should have the benefits of vaccination, not only for kids in the US and Europe and the high income countries, but, you know, this is something that we really need to make available globally. And so the CVI, the Children's Vaccine Initiative, was formed, and it really, you know, got people thinking that this is something that was important and doable. And so on the one hand, you needed to provide vaccines, but on the other hand, it was really clear that there are a lot of diseases for which vaccines could be developed, but weren't being developed, that would have a tremendous impact. So you can see CVI becoming eventually Gavi, the Global Alliance for Vaccines and Immunization, or now the Vaccine Alliance, which provides vaccines to countries around the world that otherwise would not be able to afford them. And then you had IVI, which was kind of the research and development arm, looking at those vaccines that eventually would be of tremendous use. Vaccines that were, we call them unincentivized. They're on a vaccine that a company will make, you know, $1 billion, or like Pfizer, $35 billion from the COVID vaccine. Now, these are vaccines that have to be manufactured in significant quantity at high quality and for low cost. And, you know, Gavi has this thing on their website. The package of vaccines that Gavi provides is $28, which seems like a lot when you think that, you know, 130 million kids are born every year in the world, and someone has to provide vaccines for them. The same vaccines would cost $1,200 in the United States. But these are vaccines that are, you know, have been through high quality production. The dossier, the manufacturing dossiers have been reviewed by the World Health Organization and approved by them. And that's the condition for UNICEF to be able to make these large purchases of vaccines from vaccine manufacturers around the world. So IVI plays in that universe, or that ecosystem of low cost, high quality vaccines. And, you know, it's great. Most of the vaccines that we have and that we use globally were developed in high income countries by, you know, those really innovative vaccine manufacturing companies, you know, like the Pfizer, the Merck, GSK. And then they eventually kind of trickle their way into global health. So I use this example all the time. Rotavirus vaccine, approved in the United States in 2006. By 2009, World Health Organization recommended it and approved the vaccines that were approved in the United States and Europe. So now, 15 or 16 years later, you know, with all these recommendations and with over 100 countries around the world having rotavirus vaccine on their recommended vaccine list, 60% of the world's children still do not receive 3 doses of rotavirus vaccine. So the normal pattern was, you know, it takes a while to get the vaccines onto the list and then actually to get them used. And some of the biggest problems are not the low-income countries, which get vaccines from Gavi, but the middle-income countries, which just can't afford, you know, they aren't going to get the subsidized vaccine from Gavi, a free vaccine from Gavi. They're going to have to pay for it on their own. And, you know, for a country that pays $1 a dose for this, we call it pentavalent vaccine. It's a vaccine against 5 different germs. They can't pay $20 a dose for rotavirus vaccine. It breaks the health budget. And so there have to be solutions, not only for, unknown diseases or what we call neglected diseases, but also for diseases for which vaccines already are developed. And we saw this problem in COVID. Actually, COVID had a partial solution to it, which is great. The other kinds of vaccines then are the vaccines for which no current vaccines exist and for which very little research is done. So as an example, we put $700 million a year into HIV vaccine research. This is the United States. In 2017, research on group A streptococcus, which many of us realize, it's a problem in the United States. It's called strep throat.
Sure.
You have your child has a diagnosis of strep throat. You take them to the pediatrician because of high fever. They do a throat culture or a rapid test. Within that afternoon, the doctor's office calls and says, "Oh, your child has strep throat. Go to the pharmacy. There's a prescription there, and you take penicillin, and it's done." The problem is that in low and middle income countries, often they don't take their kids to the doctor. There are no diagnostics. There are no antibiotics. And the real reason actually we treat the sore throat from strep throat is not because it makes it better all that much faster, et cetera, but because it prevents a complication that destroys the heart. And this complication, if untreated, goes on and progresses in early adulthood. So in people who are 20, 30, and 40 to congestive heart failure. Now in the US, we see congestive heart failure, people who've had multiple heart attacks, for instance. So much of the heart has been destroyed that, they have difficulty breathing and walking. They have to sit upright in bed, their legs swell. But with rheumatic heart disease, that comes on when you're young. And in women, particularly when they get pregnant, because pregnancy increases your blood volume, that makes the heart function less well. And every year it kills 300,000 people, but the US was funding $1 million a year in vaccine research, which was just not enough to get vaccines developed and tested and ultimately used. So you have to put together a coalition for these, we call them diseases of the most impoverished, DOMI, or poverty-associated infectious diseases. And it takes a while. I mean, we have to demonstrate the need for the vaccine, which means that someone has to go and get grants to do epidemiology, understanding where the disease is and how much burden it causes in countries. We know that 0.6% of the GDP of Fiji, the island nation of Fiji, is taken up with the cost of caring for people with rheumatic heart disease, this disease that group A strep causes. I mean, that's a huge amount when you consider a national economy, 0.6% of GDP. But we have to be able to show that. And having data on Fiji is great. It's a middle-income country. But what about countries in sub-Saharan Africa? Yeah. And we just don't know. So part of what IVI does and organizations like PATH are doing is really beginning to advocate for funding for these conditions. And every now and then you go to a funder like the Gates Foundation and someone says, we can just give penicillin. Not a problem. Well, the problem is we don't have penicillin everywhere. And it then becomes a problem. So understanding where the diseases are, where the burden is the start. Getting funding around a vaccine solution is the next part. And sometimes there's a company, or particularly biotechs, that have some idea, they're very interested in it, but they still need the funding to take a vaccine all the way to the end. And I think you might remember during COVID, we talked about this a lot, that it cost $1 billion to develop a vaccine, and that the failure rate from lab to licensure is 90%. So if you were going to go to Merck and say, hey, we have this great concept for group A strep vaccine that's a real problem in low and middle income countries, where you have to sell the vaccine for $1 a dose, by the way. And we really like you to get interested in it because we think this is a solvable problem. They'll look at you, they'll scratch their head and say, well, so tell us what's the business case for this? I mean, what are you really going to be what will Merck get out of it? And you don't have a strong answer because we don't know the cost of illness. We don't know the benefit of the vaccine in the United States. We don't know, whether parents in the United States would want their kids to be vaccinated against group A strep. So you have to develop a model, a business case to convince businesses that this is a worthy investment that will actually generate something of value for their shareholders. And at the other end, we have to talk to a funder. And our funder of choice is often the Gates Foundation or the Wellcome Trust, or sometimes there are countries that are interested in doing this, or other large donors. And we say, this is the case for vaccine for Streptococcus pyogenes, the group A Streptococcus. Mm-hmm. And they'll scratch their head and go, okay, all right, well, we will buy that. Here's some money to get started, and we can work in the laboratory, develop a vaccine, test it in animals, then eventually, make it in a quality manner that will allow us to test it in humans, and then take something through phase 1, phase 2, phase 3. I mean, we've heard all of this during COVID, right? Right. Oh, the vaccine's in early stage human testing, phase 1, and then phase 2, then phase 3. Oh my goodness, it's safe and efficacious. It protects against group A strep, against strep throat. Wonderful. So we take it to the Food and Drug Administration or a regulatory agency in India or South Korea and say, here's a vaccine that's safe and efficacious. Would you approve it? They say yes. Then we have to go to the World Health Organization and say, and the company that makes it does a really good job of making it. And the WHO goes and inspects the company. And often, what we do when we get funding is we work with the company to do the phase 1, the phase 2, the phase 3 testing. Sometimes the companies need support to make sure that they'll pass the WHO inspection, to make sure that everything, all the systems that they have are going to be compliant and able to be approved. And so, we'll work with them as well. I mean, this is a part of building capacity in companies, not necessarily in the United States, but in other parts of the world. And then hopefully WHO says, yes, we agree with, the Food and Drug, the Ministry of Food and Drug Safety in Korea. This vaccine is made in a high quality way. And then another part of WHO looks at it and goes, well, we're not sure it's actually going to have the benefit you think it's. I mean, strep throat is one thing, but strep throat doesn't kill people. You need to tell us that the vaccine actually prevents death from the heart disease that develops from strep throat, which happened 2 decades after the initial set of strep throats. And so unless we've planned for it, then we have to go back and follow hundreds of thousands of children who've been given the vaccine to make sure that it doesn't, to make sure that it actually prevents the illness that's killing people. And that takes additional funding. So, if we haven't planned for it, we have to go back to the funder and say, we have to have $10 million extra dollars to be able to study this for a longer period of time. And if we're persuasive, then the funder says, yes, we agree. Or if we were, if we planned well, that's actually already built into the plan. And then, World Health Organization goes, okay, it does what you say, what it does, and we agree, we'll recommend it. And then we have to convince countries to use it. But by then, we already have a huge amount of information on the cost and benefit of using the vaccine for countries. So for every dollar you invest in vaccination for group A strep, you save $20 or $40 in eventual healthcare costs. And for a Ministry of Health, that's usually very persuasive. It's got to work for the Ministry of Finance too, because in the end, someone has to pay for the vaccine and the time for all the healthcare workers to give the vaccine. And so we work to develop the formula that get a country to say, we think it might be a good idea for us to begin vaccination and then implement a vaccination program that has the consequence that we want. And so IVI has to work through the entire chain, from generating the evidence of need to generating the data that the vaccine is safe and efficacious, to working with the manufacturer to scale up production, to working with WHO to get the approvals, and then the recommendation that gets the vaccine ready to use. And then ultimately, hopefully Gavi, the Vaccine Alliance, will say, oh, we understand, we agree, this is a really important addition. We'll put it on our list of Gavi supplied vaccines. And that, again, makes it easier for low-income countries to apply to begin vaccination programs. So it's a complicated process. It's a little more complicated than in the States, where we have a COVID vaccine. There's a recommendation from the US government agent organization that recommends vaccines for use. And people say, yes, it's a good vaccine. We'll take our kid to the doctor and we'll do it. In our case, we actually have to work all along the chain to generate evidence for the cost effectiveness of vaccination. And it's something that, organizations that work in global health often have to do for medicines and for vaccines.
And on this, as I mentioned, on the DOMI list at IVI is pretty much everything. And we've talked about some of these on previous shows in terms of cholera and dengue and schistosomiasis, and the list goes on. And then I was, you just brought up COVID, and I was kind of surprised that you have at IVI a rather extensive COVID R&D portfolio as well currently. And we've discussed the mRNA vaccines. We had Peter Hotez on recently talking about the Corbavax approval. I'd love to sort of know a little bit about sort of where you're going with your COVID programs, and sort of a second part of that. We've heard this holy grail theme in the past of sort of these pan-vaccine concepts, or the concept of the universal, in this case, coronavirus vaccines. I came across this really cool paper of yours, actually, from I think it was 2016. You published with Dr. Majarad at Walter Reed you were focusing on MERS in this paper, but once again, you're working on MERS also at IVI. Any interesting stuff that you're looking at as you look broadly, say, at the coronaviruses in this pan concept as well?
Okay. Yeah. And so first, why IVI is working. I mean, and one of the questions we get sometimes is, why is IVI working on a COVID vaccine? I mean, Moderna is working on it. You have all these big Indian companies, and you have major pharmaceutical companies. You have Pfizer working on it, Sanofi, and we're partnered with Sanofi in some of their phase 3 trial testing. Why are you working on COVID vaccines? And the reason is like rotavirus vaccine. We kind of knew the innovative vaccine developing companies and vaccine manufacturing companies were going to get out there. They were going to be the ones that in 11 months had vaccines. The problem with vaccines, and we see this in global health all the time, is, the US and Europe rushed ahead and started vaccinating, where the rest of the world is still waiting.
Right.
And so, IVI decided it wasn't going to make its own vaccine. What we were going to do is help other organizations, other companies with vaccines, because a lot of these small companies can't do everything.
Sure.
So they'll come to us and say, hey, can you do the animal testing for us? Tell us if this vaccine generates any protective immune response. And then another company will, oh, well, it generated a protective immune response, but does it actually protect? Can you do a challenge? Can you vaccinate a mouse or a hamster and then show us that if we infect with COVID-19, that the vaccine is protecting? And we can do that also. Or they'll come to us and say, oh, we really want to take this vaccine forward, but how do we work outside of Korea or outside of India? Or how do we work in sub-Saharan Africa? And since we work there, we say, we can do phase 1, phase 2, phase 3. We can work with our sites in Nepal, which have really been shown to be, not very many people work in Nepal, but the investigators that have traditionally worked with IVI in Nepal have shown that they can do these phase 3 trials, not only for COVID, but for typhoid and now for cholera. You have a group of people who can do the work and are interested in doing it. And so we can help organizations with clinical trials. And then, ultimately, it was about real world evidence. And, one of the things is that IVI works with companies that aren't traditional. So Moderna didn't approach us to say, hey, why don't you do an effectiveness trial in sub-Saharan Africa with the Moderna vaccine? Janssen, Sinopharm, which is the largest manufacturer of vaccines in in China, said, hey, can you help us? And because, there isn't a lot of data around the non-traditional vaccines, around the Sinopharm and Sinovac, 2 Chinese companies, or around Bharat's whole inactivated virus vaccine. And these are companies that provide hundreds of millions of doses of vaccine every year for global health and for use in India or China. And the companies have WHO approved products, so they know how to make it at the standard that WHO requires. And so we are particularly interested in partnering with those organizations to generate data because their vaccines are widely used. And it's for exactly that reason that IVI is really seeking out some of these companies and funders that are interested in understanding how these vaccines are protecting. So that's what IVI is doing for COVID. And so these companies that we work with now in clinical testing and phase 3 testing have committed 1.2 billion doses to COVAX. You know, an IVI vaccine might not have made it through and you know, may not have found a manufacturer, but now we can say that our contribution wasn't to do something for ourselves. Mm-hmm. It was to contribute 1 billion doses of vaccine to this global mechanism to ensure that countries everywhere had access to technology for prevention of COVID. So that was our COVID philosophy. Now, universal COVID vaccines. You know, it's been difficult to keep track of all the mutations. I mean, they were, you know, between Omicron, BA1 and BA2, now we're at BA4 and BA5, and the Omicron variants are increasing in number and distribution and geographic spread at different rates in different places. So having a vaccine that works broadly against COVID-19 would be wonderful. How do we design that vaccine? You know, and again, this gets to, you know, we're back almost to where we started, not exactly where we started, because so many people have either been vaccinated or infected. But we're back where we started from a vaccine perspective. How are we going to design a vaccine against the virus that is continuously mutating? Do we have a meeting once a year, like WHO does, to pick the strains for the next, you know, for the vaccine for the next year? How far in advance do we have to do that? How do we know which ones are important? Or can we design a vaccine that incorporates features that the virus can't change? So that, you know, one of the things about viruses is and we see this with COVID, it's rapidly changing the part of the virus that allows it to spread more rapidly, which is located conveniently on the spike protein. So if you imagine sorry.
There we go.
This is so the spike protein is the part that sticks out. And additional mutations in that spike protein are making it easier for the virus to be transmitted. At the same time, it might be possible to look at the parts of the virus that are relatively conserved, that don't change as frequently, and to design a vaccine. And this is where, you know, a lot of the molecular modeling and other things are going into it. What set of mutations do we put in that will allow it to neutralize as many of the viruses that we know of so far, and hopefully viruses in the future? And do we just, you know, as some of the companies are doing, say, okay, we're going to make the Omicron vaccine, we're going to make it a bivalent vaccine. That is, we're going to include the original strain and Omicron together. Remember that the flu vaccine is either 3 or 4 strains. And we hope that those responses will protect not only against the viruses that were there before, but against Omicron and potentially for the next set of viruses in the future. And so that's one approach. So we can increase the number of types of virus in the vaccine. So multivalency, making, you know, 1, 2, 3, or 4 different viruses in a vaccine that will broaden the protective or immune responses that we see, and hopefully work against vaccines at least as far as we can see into the future. The second approach would be, again, to use molecular modeling to pick the conserved parts of the virus and try to design a vaccine that incorporates those, or conversely, to look at the mutations that we think are likely in the future, to incorporate those and see whether it whether this particular virus initially in animals protects against different new variants that you develop or you test in a computer. We call it in silico. Mm-hmm. And the final approach would be an even more kind of interesting approach, which is a pan-coronavirus vaccine, one that protects not only against SARS-2, SARS-CoV-2 or COVID-19, but against SARS-1 and maybe against MERS, the Middle East Respiratory Syndrome coronavirus, or against the seasonal coronaviruses, the one that cause a common cold-like illness. And it is possible there are parts of the virus that are very well conserved. And if we could develop this universal coronavirus vaccine. And there are actually some really interesting concepts incorporating different kinds of strategies for exposing or expressing those conserved elements and generating immune or protective responses that at this point, against what we know, look pretty broadly cross-protective. But these are mainly in animal studies now. And making the leap into relevant human protective responses is going to be really important. And that's kind of where we hope to be this year and next year, starting the initial series of experiments in humans to see whether there's protection. It's a problem though, because the way the regulatory agencies approve these vaccines, you actually have to show that the vaccine is actually preventing hospitalization, disease, and or death. But how are we going to do that now that so many people are either vaccinated or had prior infection? Thank goodness, fewer people are dying. But it also makes it more difficult because the FDA, for instance, is used to saying, oh, okay, well, you have protective responses against the spike protein. Well, some of these new concept vaccines are not going to have spike protein in them. Or maybe, live attenuated vaccines that are like the oral polio virus vaccine. They're not fully active COVID-19, but they replicate, they grow very slowly, allowing the human immune system to really attack them well and develop the kind of immune response that you would get with a good infection. The problem is that we don't have a way to license them. We haven't figured out a way other than to do a traditional large trial that looks at endpoints. So there are a number of things that we still have to work through, in addition to the theoretical concepts around these pan-COVID vaccines or universal coronavirus vaccines.
As the world has seen this, really astonishing pace, which I'll call the COVID countermeasures basket, which will put everything from diagnostics to vaccines to the monoclonal antibodies to the small molecules in there. Moving from that to your experience with some of the other nasty stuff in your career, namely the HIV and tuberculosis, major killers. Done an amazing job with HIV in terms of the triple drug cocktails. The vaccine's been a little slower. We look at tuberculosis. Well, we've had, I think we're at the 100th year of the vaccine, and we have drugs that I think were developed around 80 years ago. Any interesting learnings from the COVID work, whether it's the Modernas and the Pfizers or some of what you're doing at IVI, on how we can look back now at the HIVs and tuberculosis and speed those up as well, or maybe take some of the pace of those two killers.
So the unfortunate thing is, and this was kind of predictable actually. I was looking back over a talk that I gave 2 years ago about this is before we had a vaccine. Is it going to be an HIV? Is it going to take? Are we going to be working on it 30 years later and still not have a vaccine? And there were some things that were already kind of suggested that we knew at some point fairly early on in the epidemic or pandemic that people who got COVID were protected against reinfection for some period of time. Now it turns out that they're not necessarily protected against reinfection with the variants, but the evidence of that prior infection actually keeps the disease from becoming severe is definitely there, and vaccines add to that or can provide the protection without you being infected. The problem with HIV and tuberculosis is that the human body comes to some sort of relationship with them. It doesn't eradicate them. So, there have been a couple of cases now of people who've been reported to have spun either through therapeutic means or through natural means. Now, a couple of cases out of 40 million people who died is not a lot, but we don't understand the human protective immune response that will allow control and elimination of HIV. Similarly, again, tuberculosis goes latent, and in many people, they're infected as children, and it doesn't reappear until they're older, and then they develop very severe disease. And so the normal human immune system doesn't do a good job, that almost always makes it more difficult to develop a vaccine against an organism. But what we did learn that may help HIV and maybe tuberculosis is, we've learned how to accelerate the process of development. We've learned how to use these contract manufacturers that help to greatly scale up production of vaccine. We've learned the importance of vaccines being at a reasonable cost. So maybe those things will help us in the end. And as we look at regional manufacturing, so when you think about the case for an HIV vaccine, what is its use going to be in the United States or in Europe? The use may be restricted to high-risk groups, groups considered at high risk for HIV infection, which aren't as many people as a vaccine company would normally be interested in. In fact, while they were testing one of the vaccines, we got involved in an exercise, actually partially funded by the Gates Foundation and others, that was going to look at how to transfer that vaccine, if it was successful, to developing country vaccine manufacturers that would be able to manufacture it and take it on and actually get it out and used at a reasonable cost. And unfortunately, the vaccine wasn't shown to be efficacious. So the project stopped, but it really emphasized the point that the standard innovative high-tech vaccine manufacturers are not necessarily interested in manufacturing large quantities of a low-cost vaccine for global health. So it did teach us a little bit. COVID taught us a little bit about what it's going to take in order to get this out. And if this COVID experience manages to teach us that we need regional manufacturing, say developing manufacturing capability in Africa, as many actually companies like Moderna and Pfizer are doing, and that the WHO is focusing on, building sustainable manufacturing capability, not only in North America and Europe, but elsewhere in the world. Maybe we'll have organizations that will be able to take some of these vaccines against tuberculosis and HIV forward. I think that the other part that now we've been able to further extend our knowledge on is understanding what is necessary in order to really understand what vaccines are about. So, coming out of HIV vaccines, which is typically fairly well funded by the US NIH and the Gates Foundation and others, we were able to collect blood samples on everybody, and we can do these very detailed analyses of immune or protective responses in our vaccine trials. And that means that if a vaccine looks like it's protecting against HIV, we can go around and very quickly and at a very basic level understand what responses might have been responsible for protection. Despite, and in the hurry that we were in to look at the COVID vaccines, we didn't collect the same number of samples. I mean, in part because the studies were large, in part because they had to be done all over the world very quickly, and that we didn't have time to invest in that. But I think what COVID has taught us, again, is it's important to save samples.
Yep.
And HIV and TB have been doing that. And I think it's reinforced the need to understand at a more basic level what's important, and the importance of the right samples. Because in the end, with HIV and TB, it's likely that we will go through a process of testing. Something looks really great, go back and test it some more and make some improvements in it in order to get it to the level where it's going to be useful for global health. I think the final thing about COVID is, being able to roll things out and understand how effective the vaccines are is really a lesson that we have to learn for HIV and tuberculosis. So we go in, we do a vaccine trial for HIV, and in South Africa, it's 80% efficacious and safe, and that's great. And we go to WHO and say, we want a recommendation for the use of the vaccine. And WHO will go, well, you tested it in the setting of a phase 3 trial, which is, really wonderful science, because you test a vaccine against placebo. It really tells us, is this vaccine protecting an individual? But what we're really interested in also are, what is the cost benefit? Is it going to be useful to roll this vaccine out in every African country, or in every country in South, or in every country around the world? I mean, what is the cost benefit? go back and do a real world evidence trial, generate real world evidence that says that if we now aren't focused on the 5,000 people in the vaccine trial in South Africa, but the 40 million people in an African country, is it going to do the same thing? And if we're not ready and we don't have the funding or the information, then it's going to delay the implementation of the vaccine. And what COVID taught us is, it's really great to have evidence of efficacy. It's going to also be important to understand how this vaccine works. I mean, we still have arguments over, well, it's not really protecting against infection. Well, infection is not what matters for COVID, right? It's hospitalizations and deaths.
Mm-hmm.
And if the vaccines are doing that, they're doing their job. And what protective immune response is necessary in order to generate that? We're not really sure. We suspect it's antibody, the infection-fighting proteins that are generated by the vaccine. But for some reason, people who were vaccinated a while ago who have very low levels of antibody still are protected against death. And why is that? What are the memory responses that are coming up? So, I think understanding how vaccines work in the real world is going to be really important as we look at these global health vaccines, like against HIV and TB. And COVID has kind of reemphasized the importance of doing these and doing them right. Because in the end, WHO may not make a rapid decision on an HIV vaccine or a TB vaccine. It may want to see how it really works. And we have an example of this with the malaria vaccine, which had an original efficacy a little bit over 50%, maybe as low as 30%. But it clearly protected very young children against death. But WHO needed to have evidence that it worked in the real world, and that evidence took years to generate. And finally, last year, it recommended and approved it. So, now there's a vaccine. But in order to avoid delays for HIV and TB, we're going to have to make plans to do those kinds of trials also.
You refer on the IVI website to antimicrobial resistance as a silent pandemic. And this, we just happened to do a show a couple weeks ago with the Carbex initiative here in the States and up at Boston University. You have a program called WRAIR, the Regional Antimicrobial Resistance Data Analysis for Advocacy, Response, and Policy project that aims to catalyze a sustained demand among decision makers for high quality information and data sharing for use in global antimicrobial resistance planning, policy, and advocacy. Can you talk a little bit what you're doing on the AMR front? Because this is indeed a silent pandemic. The state of antimicrobial development is extremely poor for the last several decades. Talk a little bit about what you're doing here, if you could.
Yeah, and so it's really kind of remarkable that when penicillin was developed, the developer of penicillin said, the last thing we want is for people just to throw penicillin all around, because before you know it, there's going to be a resistant organism. And he was absolutely right. Before you knew it, within a few years, they were already recording germs that were resistant to penicillin. Now, it was an age where we were discovering lots of new drugs, and more antibiotics or antimicrobials were appearing, which was great. But we've reached a stage where these drugs are no longer coming out at the same rate. And again, there's an incentives problem. The big companies don't see a real incentive from developing the next generation of antimicrobials for use against resistant organisms. So Carbex is developed because we think that vaccines do have a role in the prevention of antimicrobial resistance. And how is that? And so this might sound odd, but we had to convince our board of trustees that antimicrobial resistance was something for IVI to pursue, because it's not necessarily apparent at first glance why it might be. But when you think about it, and there are data that, so for instance, there was an outbreak of very resistant typhoid in Pakistan. It actually spread to other places in South Asia. It was resistant to most of the antibiotics that were widely available in Pakistan, and certainly would have been resistant to the most commonly available antibiotics in sub-Saharan Africa. And so that would mean that there wouldn't be any antibiotic available to treat typhoid infection. which would be a horrible outcome. But they deployed the typhoid conjugate vaccine, these new vaccines against typhoid, and it helped to stop the outbreak. So direct evidence that if there's emergence of an outbreak caused by a resistant microorganism, for the same reason we can use typhoid conjugate vaccine as a standard typhoid outbreak, if there is resistance, the vaccine works against the resistant organism. Why? Because resistance comes from certain genes in the microbacterium, but not from the things that induce protective or immune responses. So the vaccines continue to work, where antibiotics, which stop the growth of the microorganism, don't. So that's one example. The second one is that there are data from studies in Africa, for instance, that in general, when you vaccinate for pneumonia, that you prevent and then you look to see what germs are present in the environment. The germs that cause pneumonia that are resistant to penicillin don't appear anymore. Why? Because the vaccine is preventing them from causing disease. So again, an evidence that this, we call it PCV, pneumococcal conjugate vaccine, prevents the emergence of these resistant germs is there, and we have that. The next argument is a little more indirect, that if we vaccinate against things, for instance, that cause diarrhea, that will decrease indiscriminate use of antibiotics. There's some data to support that idea. So if you vaccinate against rotavirus and cholera and typhoid, the need to use antibiotics to treat diarrhea will decrease, which will decrease the inappropriate use of antimicrobials. And that will decrease resistance globally. And that's the kind of, that's an area where additional work is necessary, but it's very complicated because we have to be able to monitor resistance using multiple different techniques. Now, does it decrease antibiotic use in a region, which means we have to have data on antimicrobial use, which means you have to go to all the pharmacies and find out, what they're selling. Or you'd have to look at resistance in the population using antibiotic sequencing of sewage water, for instance. Or you'd have to go out and collect samples from people to see what resistance genes are present in their, what we call the microbiome, which is again, very complicated kind of sequencing where we have to make some guesses about, what's happening in a person based on what we see when we do the sequencing. But, it's really an important thing. And if antibiotics can play a role, for instance, Klebsiella, most people have never heard of it. It's a pathogen that we see in the hospital in the United States primarily, or in certain populations of people who are poor and who have other medical problems. Klebsiella is one of those organisms that is on the high priority list because it has a lot of resistance, AMR, antimicrobial resistance, in the species itself. It turns out that it's also a global health pathogen. In some of the studies that are coming out now, Klebsiella figures is actually one of the major causes of death in children. So could we kill two birds with one stone, get a vaccine against Klebsiella that prevents unnecessary death in children in low and middle income countries, but also can be used as a vaccine against this microorganism in the US and in Europe that is very resistant to antibiotics. So again, the importance of research, the importance of the kind of research, for instance, funded by the Gates Foundation, that helps us to understand what is actually killing children in low and middle income countries. And is there a way to marry the two interests, the AMR interest and the global health interest? And the answer is absolutely yes, because if we can't use so for group A strep, again, going back to my favorite disease, right?
Sure.
For strep throat, we use a lot of something called azithromycin.
Sure.
In the US for people who are allergic to penicillin, or because it's broad and easy to give and fairly well tolerated. But resistance to that is emerging in group A strep. Penicillin is fine. Most doctors in the US don't prescribe penicillin, or many doctors in the US don't prescribe penicillin for sore throats. They use broader antibiotics. But if you vaccinate against group A strep, maybe that would decrease the number of visits to doctors with sore throat. It would make doctors less inclined to say, oh, okay, fine, we'll give you antibiotics for it, because we know that the child has been vaccinated. So again, it could decrease antibiotic use, decrease the appearance of this erythromycin or azithromycin resistant organism, that if it's in the United States, it's going to be elsewhere. And, you know, we don't have as many antibiotics for use in global health as are available in the United States or commonly available with the doctor's prescription. And that makes resistance a particular problem in countries that don't have full access to antimicrobial agents.
Fascinating. Really, fascinating. I wasn't thinking of the connection at first, but yeah, that's you know, clearly vaccines have a major role to play there. That's a really interesting set of initiatives. I appreciate you going through that with us. Dr. Kim, I just wanted to ask you about a couple other sort of technology baskets and just get your views on these things. One of them actually has to do with an area that I was sort of at a previous career involved in, and that was sort of this domain that was referred to in the 1990s as molecular farming, the production of various proteins and antibodies and so forth in plant systems. A lot of people are unaware. The industry really didn't take off that well, but during the Ebola outbreak, in I think it was 2014 or so, the Ebola antibodies were produced by Charlie Arnson's group in tobacco, and then, you know, I came across this paper. You're not an author on this paper, but you're mentioned in it. It was from 2013. It was entitled "Rapid High-Level Production of Functional HIV Broadly Neutralizing Monoclonal Antibodies in Transient Plant Expression Systems." And I, you know, I don't know if you were at Duke at the time, but anyway, broad question about molecular farming, and then sort of the subset of that group of technologies, which Charlie Arnson was always very hot on for developing countries. There's this concept of the edible vaccine, that we could express certain types of these vaccines, low cost. We don't have to transport them. They grow in the banana plantation or whatever. We feed them to people that need to be vaccinated, and everybody's happy. Your thoughts on sort of the state of where we are with molecular farming in 2022, and any interesting projects that you can talk about that you see on the horizon there?
So, by farming, because actually there are two ways you can look at farming, or this particular question.
This is pH.
So I think the agricultural, the tobacco or other plant-based. So there are companies that are interested in that. And actually, there's a small Korean company that has expressed interest in looking at production of different vaccines in plants. And there's a company in New Zealand. There are, you know, one of the few Canadian companies that has been involved in COVID vaccine development has a COVID vaccine manufactured. I think it's in tobacco. And of course, the weird thing is, all right, so if you want WHO to approve something, they don't want an association with the, you know, alcohol, tobacco, and firearms.
Right.
And the fact that the tobacco company partially funded this makes it, you know, problematic. But anyway, so it may be easier to use the plant as a source of, or like an organic factory for vaccines. It may not be applicable to all vaccines because, at least for instance, for HIV, there are things that plants do, not only to the protein sequence, but to the sugars that are added, that could be a problem. And the folding of a protein in a plant may be different from the folding of a viral protein that happens in an animal cell. So again, there are some things that could be technical limitations. We think that can probably be overcome. And some of the companies are working on ways to include some of those animal enzymes, those chemicals that allow the molecule to be shaped or to add sugars, in particular sugars at different areas, so that the plant can be actually genetically modified to make an animal protein, an animal-like protein. So again, I think this is really a fascinating area for future work. And, you know, some of the companies are using these supercomputers to look at the synthetic pathways that a vaccine or that a protein has to go through in order to incorporate those elements that have to be changed within the plant in order to make the right protein. So again, it's as you said, it sounds like science fiction, but in fact now there are vaccines that are being tested, one that has shown efficacy that are being made in plants. Whether this will achieve the promise of low cost, easily accessible vaccines, it'd be great if you didn't have to manufacture the vaccine in sub-Saharan Africa, but you had to grow the genetically modified plant that makes the vaccine and then take it through a very simple, still high quality, we call it, GMP process to extract the vaccine, or I guess ultimately, and actually I had lab technicians who participated in an experiment with a potato-based vaccine where they had to eat it. Maybe we'll come to that. It hasn't been as successful in humans as we would have wanted, but those are always things that might be possible. Yes, I think the molecular farming idea is a good one, but we have to further develop it and get it to scale and do the proof of concept. At this point, COVID is a more difficult proof of concept than it was. It may be important to do it with something like influenza or some other disease for which vaccines are changed frequently, and where the new concept vaccine is similar enough to the older concept vaccine that we can use similar measures to tell whether the vaccine is working. So the other part of this, which is where I thought you were going initially before you mentioned the plant-based production, is it possible to really think about all the things that we need to make in advance? And there's a really interesting program that the US NIH is trying to put forward and get collaborations for. And Danny Duick, who's one of the investigators on this, is really kind of enthusiastic when he describes this new system where they will go out. They've already identified what they think are the big, the 70 or so different diseases that could cause outbreaks in the future. And the idea is to sequence them, to understand what where the relevant immune protective responses might be, to publish them and make them available, to develop, for instance, monoclonal antibodies against some of these different things, to have frozen away so that when we need them, we can use them. At the same time, they want to collaborate with countries around the world to get samples, of people from people who've been infected with some of these organisms, so that they have what we call the reagents, the things that are necessary in order to better understand the protective responses, or to be able to develop vaccines that work more quickly, and to make them available, to publish them, to have them available for countries and people to use when an outbreak occurs. So rather than start from scratch as we did from COVID, we would have identified SARS-CoV-2 as a potential human pathogen. There would already have been theoretical vaccine constructs that might need to be made, that are published now, that are available through the collaboration, so that very rapidly we would have access to the things that we need in order to develop monoclonal antibodies or potential drug targets or vaccines against these high outbreak potential pathogens. So that's what I thought in that case, it was farming of data rather than farming of vaccines.
Farming. No, but the last place I was going to go with that, actually, and thank you, because you're segueing perfectly, is into this other concept. Once again, it's you talk about it in a couple of your papers, including the 2016 paper on MERS, but it's this topic we've heard a little bit about on past shows, One Health, and this, the connection between the humans, the animals, the environment, and as you were saying, a lot of scary stuff still out there. We don't know what's coming, but things will come. In the paper, you talk about the Hendra example, the sort of the bats, the horses, to humans, MERS with the camels and the bats. Any interesting stuff you're involved in on the One Health front, whether that is analyzing or looking for the nasty stuff ahead of time, or as in the case of things like Hendra, looking at not the human, but the animal vaccines, where there could be populations of whether it's horses or zebras or whatever it may be, that we want to try to stop it there before it does the little zoonotic hop.
Yeah, so, and again, the NIH program is designed to look at precisely this, because the high priority pathogens are ones where it's possible that there could be a leap in transmission from animals into humans. So IVI has vaccines, for instance, non-typhoidal salmonella, or some concepts around Campylobacter vaccines, which are animal pathogens as well. And we've actually had some discussions with companies that make animal vaccines. The issue has been, the level of quality necessary for production, the kinds of evidence that are required for human vaccine approval versus animal vaccine approval, and then the different animal models. So, could we take a chicken vaccine for Campylobacter and apply it to humans? And those are all really important questions as we're looking at, is this feasible? Will there be funder support for it? Because again, IVI doesn't have its own funding. We receive funding from countries, Korea, Sweden, India, Finland, and then from organizations like Gates and the Wellcome Trust. So we have to be able to interest a funder in this kind of work. But One Health is really important, and increasingly, and again, climate change fits into this, right? As climates are changing, as dry areas become wet, as mosquitoes now roam that used to be primarily in subtropical regions now are in Florida and Texas, or as areas are deforested and grazed, and we have introduction of additional opportunities for viruses in animals to transition to farming animals, and then from those animals to humans. Yes, there is definitely going to be potential. And we are working on some hemorrhagic fever viruses. They tend to be focused on Northeast Asia, which is where IVI happens to be located. But we're going to open an office in Europe actually probably this month officially, and we might be much more involved in One Health type questions that are asked in the European Union. And we're hoping that, that as we think about this, that we can really begin to appreciate not only the impact of animal to human transmission, or ping-ponging, as appears to happen sometimes with COVID, going from humans to minks and maybe back to humans, but also on the implications that climate change has for the development of diseases for which vaccines could potentially be very important. And of course, nothing happens in isolation. Climate change, animals, humans, all of this can be related in the same way that AMR is related to vaccines. Not at first glance, but as you think about the implications of AMR, or the implications of climate change, or the pressure that humans put on rainforests or forests in general, you can imagine how where we put ourselves and our domestic animals could really be important in the generation of the next pandemic.
Absolutely. It's such an exciting story, Dr. Kim, and just everything you're doing and following you is going to be continue to be exciting, and watching all the different initiatives. While I have you, before I let you go, because I know you have other things to do, but any other things going on that you want to mention to the audience, things coming up, conferences you're going to be at, talks you're going to be giving, please take the floor.
No, I really appreciate the opportunity to speak to you. And, I think where we would like to be, and remember, IVI is a development organization also. And so it's really incumbent upon us to be able to transfer technology for vaccine manufacturing to organizations that aren't in the global north, and to be able to build capacity to do research throughout through all parts of the world. And to the extent that that's doing what we need to do, then we're empowering countries to be able to help us all understand where problems are and how to prevent them. And I'm going to use as an example here, South Africa. We wouldn't have known about Omicron. Omicron appeared in South Africa, but they did the sequencing. And it turns out that they published the sequence before people in the Netherlands who had actually done some sequencing on it reported that they found it there also a transmission from South Africa. Sure. So this emphasizes, I think, and now the ongoing contributions of South African scientists to the evolution of the Omicron variants in the sub-Saharan Africa region. Again, really important to emphasize that we can't have a place where there are gaps in surveillance, because every blind spot is a place where viruses and will emerge and be a problem, and we just won't be aware until they are such a big problem that they've spilled over into the parts of the world that have capacity. So it's really important for us to remember that in addition to the vaccine and vaccination gap that exists for COVID, there was a diagnostics and sequencing gap. Oh, yeah. That really kind of helped to undermine some of the effectiveness of the vaccines that we developed early on in the pandemic. The second part is, and it's I think more a plea for, we need a new big idea. We have CEPI, the Coalition for Epidemic Preparedness Innovations, which develops vaccines for outbreak diseases. Yep. But it's important to remember that, the outbreak diseases, and thank goodness that CEPI was around for COVID, because they provided the first large sets of funding to the companies that eventually developed COVID vaccines. And MERS and Lassa fever and Nipah, chikungunya are all really important diseases. But there are other diseases that kill 4 to 5 million people a year. HIV, TB, and malaria are the big three, but we call them neglected diseases. And then diseases that don't make the WHO's neglected disease list, like hepatitis E, group A strep, Shigella. They're not considered neglected diseases, but there's no big company working on vaccines for them. But we need a way to develop those vaccines. And that means having the kind of funding that we call end-to-end. And it isn't going to be $1 billion. The cholera vaccine that IVI developed with Biotech and eventually tech transferred to companies and got WHO pre-qualified, cost the Gates Foundation end-to-end $28 million. This was not $1 billion vaccine. There you go. The typhoid conjugate vaccine will cost the Gates Foundation $29 million. So we're not necessarily talking about billion-dollar funds for the development of these high burden endemic diseases of poverty, but a fund needs to be developed. And really, if we could get consensus, if we could get a big country, a G7 country, to say, hey, this is something that we would like to take forward, we would be really eager to partner with them to develop the concept for this kind of neglected disease vaccine development fund.
It's fascinating. And as I said before, I'm wishing you the best with all of this, not just in the development work, but in the advocacy and spreading the word about the need and these gaps, and really and making us more aware of the current situation and where we need to be going. So a really exciting journey. I'm really honored you took the time to share it with us. For everybody that's going to be listening to this episode on the podcast networks or watching on the YouTube channel, again, you've been listening to Dr. Jerome Kim, Director General, the International Vaccine Institute, discovering, developing, delivering safe, efficacious, affordable vaccines around the world for major global public health needs. Again, Dr. Kim, thank you so much for taking the time out of your schedule to come talk to us for a little while. Obviously, thank you for everything you're doing there at IVI. And as we like to say on this show, thanks for helping to create a better tomorrow for many people through what you're doing. It's really a fascinating story.
No, thank you for interviewing me and for this podcast, which I think will advance global health in multiple ways. Thanks a lot.